Hello, and thank you for joining the Pituitary Network Association's webinar program today. The PNA is dedicated to educating people with pituitary disorders, their families, and their healthcare providers. The PNA is a nonprofit organization that relies on the support from our members and donors. During the webinar, please feel free to type in your questions at any time. Note that all questions will be saved until the end of the webinar. We have allotted time to answer as many questions as possible. Today's webinar, Intracellular Glucocorticoid Reduction, an investigational approach to management of Cushing syndrome, autonomous cortisol secretion, and the side effects of glucocorticoid medicines, is presented by David Katz, the founder and chief scientific officer of Sparrow Pharmaceuticals, a clinical stage biopharmaceutical company developing novel targeted therapies to address unmet needs in both endocrinology and rheumatology. Please hold, as there will now be a brief delay as we change presenters.
Sorry, I had just been remuted. I'll start again.
Okay.
Thank you for that nice introduction, Danielle. This presentation will focus mainly on posters that we presented as a company at the European Congress of Endocrinology, the Endocrine Society meeting, and the European League Against Rheumatism annual meeting during the last month in Milan, Copenhagen, and Atlanta. This presentation discusses an investigational drug. It is not for promotional purposes. SPI 62, the molecule that will be discussed, is presently in phase 2 clinical trials for patients with ACTH-dependent Cushing syndrome, autonomous cortisol secretion, and in combination with prednisolone, polymyalgia rheumatica. And I will discuss at least the Cushing's trial later in this presentation. So there will be some switches because I'm going to be sharing the various posters and talking over them sequentially. And so let's see if this technology works here. Nope, that's not what we wanted. There we go. Okay. So recognizing that this is a pituitary-focused organization, I will have to tell you at the outset that the pituitary and its role in the hypothalamic pituitary adrenal axis is only half of the story of cortisol and the diseases that are associated with endogenous excess of cortisol, Cushing's syndrome, and autonomous cortisol secretion. And we went for about 50 years since the characterization of Cushing's syndrome by Dr. Cushing back in the 1930s, and over 30 years from the discovery and initial medical use of cortisone at the Mayo Clinic, before we realized that there was a second important metabolic pathway involved in cortisol biology. And Of course, the canonical HPA axis is an important part of cortisol biology, but what was started to be recognized now about 35 years ago is shown in the figure at the middle left of this poster from the Endocrine Society meeting. And that is, when you think about cortisol and cortisone in the bloodstream, similarly as we think of drugs as pharmacologists, you realize that what's the important fraction of cortisol and cortisone for biology is what can actually get into cells, because the mineralocorticoid receptors and the glucocorticoid receptors, even those that are membrane bound, are all inside the cell. And so the molecules have to get inside the cell to act. And while in general circulation, the ratio of cortisol to cortisone, at least in a healthy adult, is about 4:1, cortisol is much more bound by cortisol binding globulin and albumin than is cortisone. And so if you look at it in terms of free fraction, it's actually much closer to equal. And so quite a bit of cortisone can get into the cells, and that's represented in the figure here. And there it's transformed by an enzyme called 11-beta-hydroxysteroid dehydrogenase type 1, or HSD1, into the active form cortisol, which can then interact with the receptors. And HSD1, as shown in the figure at the lower left in this poster, is part of a 2-enzyme cyclic metabolic pathway that affects cortisol. So the inactive forms of glucocorticoids, whether cortisone or prednisone or other inactive glucocorticoid, or the other inactive forms of the other glucocorticoid medicines, are transformed to the active forms such as cortisol and prednisolone in tissues in which ordinary levels of cortisol have physiologic functions. Excess levels of cortisol can have pathologic functions, and those include organs such as adipose, liver, bone, muscle, skin, eye, and brain. In other tissues, such as the distal tubule of the kidney, which requires protection from cortisol to prevent its mineralocorticoid activity, there's a reverse enzyme, 11-beta-hydroxysteroid dehydrogenase type 2, or HSD2, which inactivates cortisol, prednisolone, dexamethasone, etc., to inactive forms. In the liver, there's an additional metabolic pathway that's an excretory pathway that involves the 3 and 5 steroid reductases, and those enzymes, sorry, we're having technical difficulties here. Those enzymes convert cortisol and cortisone to metabolites that are rapidly excreted in the urine. And so a good part of our work relies on the measurement of those metabolites in urine, and actually leads us to think that some measurement of those metabolites might be a better way to monitor Cushing, the status of patients with Cushing syndrome or autonomous cortisol secretion than is urinary free cortisol. And so one way that we use these is by the ratio. So if you look at the sum of cortisol metabolites divided by cortisone metabolites, that gives you an idea of the activity of HSD1 in the liver. So the pharmacologic effect of our drug, which is a potent inhibitor of HSD1 and is highly selective against HSD2. It's at least 3,000-fold more potent for HSD1 than it is for HSD2. And so what you can see in the very upper left panel of all these graphs is the HSD1 ratio, this urinary ratio, and the effect of SPI62 on it, both after a single dose and after multiple doses. You can see that that ratio is reduced by about 90%. The components of that are shown in the three sets of graphs to the left of that. So the left two panels show the two cortisol metabolites, each of which are reduced about 50% by SPI62. And the third panel represents the cortisone metabolite, which is substantially increased, which you'd expect a buildup of the substrate cortisone when the enzyme is blocked. And what these data indicate is that about 50% of the intracellular cortisol, that is the cortisol that can access the receptors in the hepatocytes of healthy humans, is made by HSD1 and does not come directly from the adrenal. Now, about 15 years ago, there were some clinical pharmacology studies that were done using labeled tracers of cortisol and cortisone. And those studies established that on a total body basis, that close to 50% of the total body cortisol production during a day, again in healthy adults, comes from HSD1 in the splanchnic bed, i.e. the liver and abdominal adipose, rather than from the adrenal, and our data are consistent with that. Now, the effects of a potent inhibitor of HSD1 on the endocrine system are rather interesting. And there's one observation that stands out in particular, showing how much effect just changing intracellular cortisol can have on systemic cortisol. And those are shown in the fourth row of figures on the poster, that there are, and these are serum cortisol measurements, placebo compared to SPI62 at various time points, either after a single dose or after multiple doses. And focus particularly on the second set of panels, which is 2 hours after a single dose of SPI62. What you can see is that there is about a 1/3 decrease of systemic cortisol, owing only to the inhibition of production of intracellular cortisol. The row of figures immediately above those for serum cortisol or for plasma ACTH. And what those show is that at that same time point, 2 hours after a single dose, the HPA axis is still asleep. It thinks all is normal. But by 4 hours after the first dose, the decrement of serum cortisol has removed negative feedback sufficiently that the HPA axis wakes up. The pituitary produces about 50% more ACTH. It continues to produce that approximately 50% more ACTH throughout dosing with SPI62, whether you look during the day or in the evening. And that restores systemic cortisol homeostasis. So at time points 12 hours after the first dose or any of the time points after multiple doses, you can see that the serum cortisol levels are the same in healthy adults who were exposed to SPI 62 as in those who were exposed to placebo. Secondarily to the increase in ACTH, there's also an increase in androgens. So that's shown in the very bottom row of this poster. Here, blue symbols are males and red symbols are females. And you can see this for DHEAS and then separated by sex for testosterone. There is a modest increase of androgen precursors. What's true for DHEAS was also shown for DHEA and androstenedione. And in females, there is also a moderate increase of testosterone. To date, with SPI 62, as well as with other HSD1 inhibitors for which data have been published, in general, androgen levels, individual androgen levels stay within normal ranges, and there have been no associated clinical consequences that have been clearly noted. For example, there haven't been any substantial number of reports of menstrual irregularities, hirsutism, acne, etc. So that's what a HSD1 inhibitor does to the HPA axis. And now I'm going to switch gears and get into the question of, well, so what does this have to do with Cushing syndrome and other disorders? So I'm just going to stop sharing for a moment. And I am going to switch slides. So all right, so the story started about 20 years ago with the presentation by Tomlinson et al. Of an unusual case study, which is this woman who is shown on the right. She presented with an androgenic side effect of amenorrhea. And on endocrine workup, it was discovered that she had very high levels of urinary free cortisol, and so should have had Cushing syndrome, but she didn't. It turns out she also had a condition known as apparent cortisone reductase deficiency, which is an enzyme deficiency of HSD1. And so without HSD1 to convert inactive cortisone into cortisol in target tissues for cortisol morbidity, she had none of the glucocorticoid related symptoms of Cushing syndrome. Subsequently, her pituitary tumor was found on imaging and was removed, and then she received a course of hydrocortisone replacement after surgery, and there's not a lot of information. There's not information available on her clinical course after the initial hydrocortisone replacement. There was a second similar case study from Japan that was reported a few years after. And so what this suggested is that perhaps if HSD1 is not active, one could block or maybe even reverse many of the symptoms of Cushing's syndrome, and that is the approach that we're taking. So after these initial 2 case studies in 2014, the phenotype of the HSD knockout mouse was described. When given exogenous corticosterone, which is for rodents the main glucocorticoid, the knockout mouse showed none of the glucocorticoid-related symptoms of Cushing's syndrome, where control mice did. And I'm about to switch again to the poster that we presented at the recent rheumatology meeting, which shows that we've recapitulated that result with our inhibitor. Okay, so in this study, mice were administered corticosterone in their drinking water for 5 weeks, and then they were either given vehicle for SPI-62 or 3 different doses of SPI-62. And what we observed is that on food consumption, weight, cardiometabolic, muscular, and dermal adverse effects of corticosterone, that SPI-62 prevented those in a dose-dependent manner. So it's probably under the adage that a picture tells 1,000 words, I think the skin is the best place to focus first. So these photographs that are in the middle of the poster, you can see the normal dermal architecture and thickness in the upper left control photograph that is substantially disrupted by corticosterone by itself. And then in the lower row, you can see the dose-dependent restoration of pretty normal dermal thickness and dermal architecture with increasing doses of SPI-62. Similarly, food consumption was increased by corticosterone, and then in a dose-dependent manner restored to control levels by SPI-62. The effect of corticosterone on weight in this study was biphasic. In the initial period, there is a decrease of body weight gain associated with corticosterone, and that was not affected by SPI-62. But then after about 2 weeks, the mice start to have accelerated weight gain relative to controls, which is more analogous to what happens in humans when we receive glucocorticoids. And you can see that that was prevented in a dose-dependent fashion by SPI-62. In the table on the right, these are preliminary analyses at the time that this poster was submitted. The formal analyses of the data were not yet available, so we just looked at descriptive statistics at the mean change compared to the control group in port-treated mice. And what you can see is that for example, on HOMA-IR, there are quite large increases of that in corticosterone treated mice, but in a dose-dependent manner, those increases were prevented by SPI-62. And similarly, for body fat content and body muscle content, which were measured in an echo MRI total body composition analyzer, and as well then at post-mortem evaluation, the weights of various fat depots and muscles, and also while the animals were still alive, corticosterone caused a decrement of grip strength, which was reversed and maybe even improved by SPI-62. So having all this information, we're now going into clinical trials, and I will switch once again. Just a moment. To describe what we are doing in ACTH-dependent Cushing's. Okay, so this is another poster from the Endocrine Society meeting, and this is a study that will enroll. It's a proof of concept trial. It will enroll up to 26 subjects with ACTH-dependent Cushing syndrome, so either pituitary or ectopic disease. Enrollment is planned to continue until we have at least 18 subjects enrolled with pituitary Cushing's, and the main analyses of the trial will be on the patients with pituitary Cushing's only, but then a sensitivity analysis will be done both on the more limited number of patients with ectopic disease, as well as the total study population. Patients will receive different sequences of SPI-62 and placebo for 12 weeks each during a 24-week treatment period. And they will undergo quite a few procedures to measure the adverse effects of cortisol in the body. Aside from quite a bit of laboratory testing to assess various biomarkers of things like cardiometabolic and bone health, they'll also undergo an oral glucose tolerance test to look at the effect of SPI-62 on excursions of postprandial glucose, which is the main glycemic effect, or a main glycemic effect of cortisol excess. We'll also be looking at continuous glucose monitoring, both during and proximal to the OGTT, in the hopes that we might be able to use CGM in the future to be a less invasive, less inconvenient way to monitor glycemia in patients with Cushing syndrome than is the oral glucose tolerance test. Patients will also undergo DEXA scans and 24-hour ambulatory blood pressure monitoring. Again, one of the main effects of cortisol excess on blood pressure is the loss of the nocturnal dipping phenotype, which can't be captured with office-based with office-based measurements. In addition to the ABPM, we will also have trial participants take their blood pressure at home in the morning and in the evening, again in an attempt to find a less inconvenient way than 24-hour blood pressure monitoring to capture the nocturnal dip phenotype in patients with Cushing's. Besides being randomized to different sequences of SPI-62 and placebo, so every subject again will receive both for 12 weeks. There will also be 2 different time points at week 12 and week 18, and subjects will be randomized to one of those 2 visits, but not both, to reduce both site and participant burden. The trial will be run at various sites in the United States, as well as in Bulgaria and Romania, and we are still actively looking for additional investigators for that trial. Besides the trial in ACTH-dependent Cushing's syndrome, we also have a trial that has started in patients with cortisol secreting adrenal adenomas, and that trial will include patients with autonomous cortisol secretion as well as adrenal Cushing's syndrome. And then, as I mentioned earlier, in combination with prednisolone, we're looking at the ability of SPI-62 to block some of the side effects of glucocorticoid medications. So that's a brief introduction to our approach and our program, and I will be happy to take any questions that you have.
Thank you so much for that very informative presentation. We do have one question that just came in. Now's a good time if anybody else has a question, go ahead and send it over. Our first question is, where does a patient go to join the trials?
Okay, so let's see here. Well, so the best place to look is on, so there are 2 places to look on the web. So one is on clinicaltrials.gov, which gives a sense of where the study centers are. You can also look on the company's website, sparrowpharma.com, for additional information, and there's a way to contact the company directly on the website if a patient is interested in the trial and wants to learn what is the closest center to them.
Okay, thank you. Next question. I had Cushing's. I'm not certain that I still do. I also had 2 surgeries. How will I know if I qualify?
Okay. So as many patients, Cushing's for you appears to be a very long journey, and it's exactly the sort of patient who's had surgery, perhaps more than one surgery, and has tried medical therapy and still has symptoms that are related to Cushing's, that their Cushing's isn't well controlled either by surgery or by available medicines, that are eligible for this trial. And so I would encourage you to go to our website and get in contact with the company, and then in a private manner, can go over some more of the some more information about your particular history to learn whether you are a good candidate for the trial.
Okay, thank you. We have a few comments. Great info. Thank you very much. Very exciting news. Great presentation. But we do not have any more questions. It was very thoroughly covered and very informative. So we really appreciate you taking the time to do this today.
Yeah, and I'm glad to have heard from a patient, because what we're doing is all about the patients. So how can we make your lives better?
Yes, definitely. Well, we thank you, and this will conclude today's webinar presentation. We appreciate everyone taking the time to join us. If you missed any part of this webinar, or if you would like to share it with family members or friends, it will be available on our website at www.pituitary.org after it is edited. There will be a brief survey after the webinar. Please feel free to fill it out, help us provide you with the information you need. And we thank you again for joining us, and enjoy the rest of your day.